Hello everyone, my name is Mohit Kumar from group 317. Today my topic is transcription. What is transcription? In transcription form there are mainly RNA. In transcription there is a two strand, 3' to 5' and 5' to 3'. Only one strand take part in transcription. Both strands do not take part in transcription because if both strands take part in transcription, it form a complex structure which never form the proteins. This is the overall structure. The 3' to 5' is called as template and non-coding, whereas 5' to 3' is non-template, which is also called as coding. Here is the promoter region, this is the structure region, here is the terminator region. There is an enzyme which is called as RNA polymerase enzyme, which goes to the promoter region for starting the transcriptions. And at the end of transcription, the row factor is coming which bind to the terminator region and the transcription will stop. Here's the full diagram of transcription in prokaryotes and eukaryotes. First, in prokaryote cell. In prokaryote, the enzyme present is RNA holo enzyme. It is a core enzyme which contains 2 alpha, 2 beta and omega. Here is also a sigma unit present. It's form the rRNA, mRNA, tRNA. If rRNA form, which is mainly ribosomes, which also help in translations. If RNA polymerase 2 form the mRNA, it is a modification of transcription. If the third is the tRNA, which is mainly for the transfer of amino acids which help in translation. Mainly there are three RNA polymerase enzyme: RNA polymerase 1 which form the rRNA, RNA polymerase 2 which form the mRNA, RNA polymerase 3 which form the tRNA, and RNA polymerase 2 or 3 both form the snRNA. Here's the three steps of transcription. First is initiation. In initiation, in prokaryotes, initiation there's two regions present, -35 regions and -10 regions. In eukaryotes, it's contain the Tata box, CAT box and GC, which is used in RNA polymerase 2. Second step is elongation. In elongation, RNA polymerase that read the template strand. It open the DNA, stabilize the DNA, and then the synthesis the RNA. It's reach the 5' to 3', which is also called as proofreading. At last, the third step is termination. In termination is two type: row dependent termination and row independent termination. If the row factor bind to the terminator region, it is called as row dependent termination, whereas if row factor doesn't bind directly to terminator region, indirectly bind to terminator region, which is called as a row independent termination. This is in 5' to 3', there is a capping or tailing occur. In 3' mainly tailing occur, in 5' mainly capping occur. In capping, the enzyme present is the methyl transferase which help in capping, which initiate the transcription and also prevent the degradation of transcription. This is the immature RNA. Immature RNA contain both exon and intron. The splicing was occur by the synripse enzyme. It's in splicing, it's remove the introns. Introns are removed completely and only exon left. We can see hnRNA, which is a immature RNA. In splicing, it form the mature RNA. Here's the difference between coding strand and non-coding strand. Coding strand also called non-template, whereas non-coding strand also called as template strand. The coding strand determine the correct nucleotide sequence of mRNA, whereas the non-coding strand act as base of mRNA in transcription. Coding strand is also known as plus strand or sense strand, whereas a non-coding strand is also known as anti-sense strand and minus strand. Coding strand do not take part in transcription process, whereas template strand take part in transcription, help in formation of mRNA. The coding strand has complementary nucleotide sequence like 5' to 3', 3' to 5', whereas the template strand do not have the complementary sequence. In promoter regions in eukaryotes, there is a box called as Tata box. In prokaryotes, this box is called as Pribno sequence. In eukaryotes, it's called as Tata box. My second topic is about RNA and their types and their transfer. The first RNA is ribosomal RNA. Ribosomal RNA is the most abundant RNA, total 70 to 80% of total RNA. Ribosomal RNA further divided into 23S and 28S RNA. RNA is a constituent of ribosome. Depending upon their sedimentation coefficient, RNA is further divided into 28S RNA, 80S rRNA, 5.8 sRNA and 5 sRNA. rRNA in prokaryotes, which is 3, mainly 23 sRNA, 16 sRNA and 50 sRNA. Second is tRNA. tRNA is also called as soluble or sRNA, in which it is known as before the discovery of genetic code. There are 100 type of tRNA. The transfer of RNA constitute about 15% of total RNA. This is the structure of tRNA is contained that there is a codon which is 5' to 3' and anticodon. This is a detailed structure of tRNA. In 3', this is the site where the amino acids are attached. This is the codon site and it's complementary. There is an anticodon site which form the vowel position. In also is show the T loop and D loop. This is a amino acid binding site which is 3' and T loop, D loop, and next is codon anticodon loop. And third type of RNA is called as mRNA, which is a messenger RNA. This is also called as genetic RNA and informational RNA. This RNA further goes to the translation process which occur in cytoplasm in both prokaryotes and eukaryotes, which form the proteins. In transcription, it's from RNA, and RNA to protein formed by the translation. So this is how the tRNA help in translation process. This is whole structure. Thank you very much.